Thanks for coming. We are pleased to introduce Dr. Rodrigo Pavão from the Federal University of ABC. Today we will present neuron computational principles of sound source localization. Please join us in welcome Rodrigo.
Thank you. Nice to be back here. Thank you. Neural and computational principles of sound localization. That's the name of the talk. So it's about checking where the sound comes in all its dimensions, right? Azimuth, elevation, and distance. And when we talk about azimuth, we have two main cues, right? That are the important. We compute the location of the sound in the azimuthal plane using level differences and time differences, right? So ILD is the cue, right, for level. So you have a shadow of the sound. So the sound source is here. It arrives here with louder here and lighter here, and this difference in level is the dBs there. This works. Well, no. So you can get compute the location of the sound from the level difference between the ears, right? And there is a circuit for computing the level differences involving the LSO, the lateral superior Alves, and so the sound coming from left goes to LSO through excitation and goes to LSO coming from the opposite side as an inhibition. And this balance between excitation and inhibition is used for computing the... thank you. Funciona o laserzinho? A bolinha vermelha? So, and this balance here allows to get the firing rate that leading to the perception of left and right. The other cue, the one that we'll center more, we'll talk more in this talk, is the time differences, right? So it's in the microseconds scale, and it's pretty redundant with the level. And the time differences are very redundant with the level differences. But there is a different circuit, and it's also with excitation and inhibitions, but there is two excitations and two inhibitions, and I will talk more about this later. This will be the main topic of the talk, discussing ITDs, the processing of ITDs. But there are also, as I said, there is this azimuth, elevation, and distance. The elevation is coming from the shape of the ears, so it gives a signature in the sound that reaches your ears. There is a signature on the spectrum, right? So with some valleys and some tops of the hills, right? So and there is cells that respond to all of these, right? The experiments were done in cats, performed in cats, and the cats also have this different signatures depending on the elevation of the sound. So here is the sound coming from the midline, right, from the front on 0 degrees. If you change going to top or bottom, right, there is a different signature, and you have cells that respond to these peaks and troughs, right? So it's like from vision is analog to the on-off cells, right? Center on, center off. So you also compute from this, right? And also distance, you use level, reverberation, spectrum, all of this to compute the position of a sound source, right? The focus here is on time differences, right? As I said, so the ITDs, right? The interaural time differences. And the big picture of this talk is to link the psychophysics to physiology in this context of sound localization, right? So here is a sound on 45 degrees, or 45 degrees on left, and the sound reached the ear a bit earlier on the left than in the right, right? And if you go to the other side, you have a different time difference. And this is known for a very long time, right? So in the early, in the last century, first decades of the last century, there are engineering mechanisms to be able to locate from where the shot was coming, right? Something that is used on war, right? So the idea was to compare the sound that comes from different microphones, right? Microphones on the left and right, and you check these differences in time, you align them, right? And when you align the sound correcting this lag, you have an estimation from which location the sound comes, right? So it's pretty straightforward maybe there is something in the brain that is doing something like that, right? And this is the first model of the computational neuroscience, theoretical neuroscience back in the 40s, right, of the last century. And they proposed this model, right, that is the sound coming is an engineer, right, that has some concepts of neuroscience. They said, neurons could compute which it's the lag of the sound as done by this equipment if different cells were excited sequentially, right? So the sound coming from left would excite first the neuron A, then the neuron B, then the neuron C, sequentially, right? So the sound from the left would reach all those neurons sequentially. And the sound coming from the right would follow this, would stimulate also the same, would excite the same neurons but in the opposite order, right? And so with this difference, these delays, different delays, and some coincidence detectors, right? There is a stimulation from both sides at the same time. Would be different cells would activate to different locations. This is the classic place code coding, right? Place coding. So each neuron coding for a different location in azimuth using this time difference Q. Okay? So the neuron F would be coding for location minus 45 degrees, and the neuron C would be coding for the neuron at 30 degrees. Right? Very beautiful, but is there any physiological evidence for this, right? And in the 80s, they found exactly this circuit in the owl, right? So became something super interesting, right? So we have a proposal from the theoretical neuroscience that is matches exactly the electrophysiology results from the brainstem of the owl, right? From the here the nucleus laminaris is this sequence of neurons there, and this was also adapted for humans, right? So there is there is no recordings specifically from this system from humans, but the idea was to adapt what is proposed was proposed and found in owls, and check if this is also present in humans, right? It came from but it started with a psychophysics approach, right? So here is a protocol also from the 50s, and in which a person is sitting on an anechoic chamber, and the sound is coming from a speaker, and the experimenter could change the location of this speaker, and the sounds were presented in different frequencies, right? And the idea was to check how much it has to change to be able to detect a difference, if this sound source was moved to the left or to the right. So a sound coming from the front, so the reference ITD is 0, right? So it's right in the front, the azimuth 0, right? And that it has a ITD of 0 also, right? So in this location, the thresholds are very, very low, right? So it's around 10 microseconds. But if you go more and more to the periphery, around 800, so it's stream left, right? Stream left or stream right here, and the thresholds are much higher. And the idea was, okay, how we have to change this model to capture these properties, right? To match what they find, they expect to find in physiology, and to adapt, to match what they find in psychophysics. And the proposal was to include more and more neurons responding to the front. So there is a higher density of neurons responding to locations, to ITDs near zero, right? And if you, the model was not created exactly for this, but if you put this density of neurons as proposed by this model as a function of the threshold ITDs, you see that there is a monotonic function, especially at lower frequencies. If you exclude these high frequencies, you have something like monotonic, right? Something that with more density of neurons, you see a lower threshold, right? So everything is matching in this idea, right? It also matches the amount of information, right? If you use a concept like Fisher information, right? That there is, you're trying to estimate an azimuth by the measures of the time differences, right? You have a higher slope on this association between azimuth and ITDs, right? In this higher slope would be the range where the azimuth can be estimated with lower uncertainty, right? So this is the Fisher information approach. So it has more information about the ITDs have more information about azimuths in the frontal region. So everything matches, right? Everything makes sense, right? This model designed by engineers is supported physiologically by owls, supported by information measured by the slope, and predicts well the psychophysics data. Everybody happy, everybody that built this, and Darwin and Fisher, Ronald Fisher with this data from psychophysics and electrophysiology from mouse. But then it's not what has been observed in the recordings in mammals, right? So a group from McAlpine recorded in guinea pigs, and also they made these recordings in gerbils, is another rodent. And what they see is that when you record on colliculus or the MSO, what you see is that all the cells here is each line is a different cell, a different neuron, right? So it's they were presenting sounds on the earphones for the guinea pigs, and they checked. They presented white noise as a stimuli in different ITDs. They presented sounds that went well beyond the ITDs that the animals listen in the environment, right? So this is the physiological range of those animals, right? So it's a small head. So the difference, the ITDs that they here is about minus 150 to plus 150 microseconds. But they presented sounds that have a ITD that went far beyond that. But what they see is for those neurons that are tuned mostly to frequencies near 250 hertz, is that all of them have the same shape. And this is observed for all the frequencies that they tested. So you could do something like an average, right? From all the cells here, you have something like an average. And you definitely see something that doesn't match at all that other proposal, right? That has been observed in owls. So that place code model doesn't match much, doesn't fit the experimental data recorded in rodents. And so probably what was happening here is that the ITD is computed using rate, right? So different in rate, the firing rate of the neurons are coding for the locations, right, for the azimuths. And in this sense, the higher slope here will be the higher feature information. So will be the locations that will be easier to so for higher firing. The higher slope here, we'll see it from the firing rate to code for the ITD easier. To estimate which ITD is presented from the firing rate. So it will be easier to check, to decode which ITD it is, right? To estimate which ITD is from those firing rates. And interesting also is that this is always ITD as a function of the firing rate. But if you, instead of putting this difference in time, you check this in phase, right? So it's just normalizing to the frequency. What you see is that all the neurons are they are tuned to the same IPD, to the same interval phase difference, the difference in phase, right? So it's definitely not the same model, the place code model here, right? And finally, I'm presenting here how is proposed to do this this coding in rate. So MSO would receive excitatory and inhibitory inputs, right, from the left here, for example, and this is orchestrated in time in such a way that there is a excitatory, then inhibitory postsynaptic potential, right? So it has a pulse, right? And when you have a stimulus that is presented only on right, what you have is the opposite, something similar, but starting with inhibition, then excitation, right? And the principle to compute a firing rate from this sounds varying on ITD, right? So the left comes earlier, or the two at the same time, or the right comes earlier. From this ITDs would be a sum of those potentials, right? If the peaks are aligned, there is a high firing rate. If they are aligned oppositely, so the excitation is aligned with the inhibition, there are no spikes, and so you don't have any firing rate. So this allows to create a curve like this, relating ITDs to firing rate. Okay, makes sense? And so then it comes this computational approach. What is happening there? What is happening that is so different from what you see in rodents and what you see in owls, right? There is a group that decided, let's do a simulation here in which we put we have the tuning curve as a function of IPD, right? The firing rate as a function of IPD, and you have the peak here. So the feature information, how much you can estimate from the IPD based on the firing rate, will be higher where the higher slope of the tuning curve is, right? So this red curve here has this M-shape levels of feature information, and the simulation, they put all the neurons spread, and they are trying to optimize in the physiological range. So those are around this triangle here would be the IPDs that are physiological to the animals, right? So to this here are for the gerbils, and they are they are tuning, right, the center of these tuning curves, and they are shift in order to maximize the feature information in this region of this triangle. And this is the optimal coding for this, for the optimal coding, the optimal tuning for the IPDs to code for those physiological IPDs will be in this way. So it's one group of cells tuned to one side, another tuned to the other side, exactly as seen here, right? All to the same IPD. So this will be the explanation. And for owls, it will be a bit different, because they have a physiological range that is, the IPDs that they listen in the natural environment, it's much, they are tuned, they are able to detect ITDs in much higher frequencies, so it will be more spread distribution, right? So this will be optimal, the optimal coding of ITDs in the physiological range will be in that way. And this, it comes what would be what would be expected for humans, right? So it's putting the same triangle, the Fisher information will be like that, right? So then the attempt is to check if the psychophysics are able to match these information values. And there is no function that connects those well. So it comes with this issue, right? There is the field sees, there is a group of people working with this place code model that has no physiological support. And there is this rate code model that has no psychophysical prediction. And the idea was to conciliate these inconsistencies, right? This is a paper that I published in 2010-20. It was not well understood at that time and not very cited. So I'm making a review now, trying to explain this in a clearer way. And the idea is that the ITD is not a constant, is rather a distribution. So it has a mean and has a standard deviation. And this acoustic Fisher information, how much you can estimate locations based on ITDs, can predict both psychophysics and electrophysiological results, right? So this is the basics of how we are computing the ITDs, right? So we take a signal coming from a location, filter as the cochlear filters, and compute the instantaneous ITD, and we extract a mean and a standard deviation of those, right? And from this mean and standard deviation, right? So here is the same graph that we saw before from the other older figure, classic figure, but there is also this other metric that is standard deviation, and from those we can compute the Fisher information, right? And here is the Fisher information and a color plot too. So it's higher information in the front and lower in the periphery. Across all frequencies, you see higher information of azimuth in the frontal ITDs. And these ITDs are, when you try to explain, to check the correlation between those ITDs and this Fisher information, you see that there is a clean relationship between them. So you are able to predict the thresholds of psychophysics using this acoustic Fisher information. So this is how much information is the sound that reaches your ear have of information about azimuths, right? And the other part from psychophysics, from electrophysiology, is we took that same model that they were running here, right? This approach of higher Fisher information, but of maximizing Fisher information in the physiological values. But what we try to do is let's match the Fisher information acoustic with the Fisher information of the population of neurons, right? So a lot of neurons trying to estimate, to have the same Fisher information values, same organization here, you see that it's very well correlated. So it gives a limit of amount of information, neuron Fisher information that matches the acoustic Fisher information. And when we do the simulation to have this curve, to check this heat map in the same levels as those, what we see here is those higher density of neurons in one side than the other, right? Exactly as seen in the results from the Goebbels, right? So this is the idea that we are able to predict both psychophysics and electrophysiology with this approach, right? So now we are running, we replicate all these results. We did new simulations and got the exactly same results, and we are trying to get reproduce the same analysis in the same species, right? So taking the data from Goebbels, or maybe get some data from other groups, marmosets maybe, but at least the theoretical part is done very well. We need this new data, right? And that's it, right? So what we are doing now is, with this my collaborator, Boris, the last picture there, we are working a lot there in UFABC, and we are working with all these approaches, right? Using acoustic analysis, statistics, natural history, psychophysics, data from electrophysiology, and putting all together to explain sound localization, connecting psychophysics and electrophysiology and neurodata. And that's it. Thank you.
Thank you, Rodrigo. We're moving to the questions.
I was wondering, because in the textbook knowledge always cites these models now of the interaural difference for sound localization, and that there's a difference when it comes to the higher frequencies and the lower frequencies, because the higher frequencies you cannot use phase differences, you can only use the temporal differences. Can you comment about that? And the other thing is that I remember the superior olive as a point to record from, or where the first binaural interaction is. Can you also comment on that?
Yes,
while
yes.
you're in the colliculus?
So this model with LSO and MSO, it's what's presented on the textbooks like Kandel. It's the LSO model. Is great, but what they are showing in for time differences is something like that. So this is from OWL, and this is what is in test books still today. You check the last version of Kandel. What they are showing is this. So it's taking the circuit that is observed in owls, and they are putting in the olives. So it's very inconsistent with what? Yes, yes. But it's putting the circuit that is found in one animal in humans, for example. They are putting, they draw the human brain with this circuit there. And what is, there is much more evidence now is something like this, that has inhibitions also, right? And the circuits that are shown. Yes, yes, there is this, for mammals, what you see is when the hair, for the hair cell, when there is a phase locking between the hair cells, the oscillation, the superficial oscillation of the hair cells, they are not, what you see is that this phase locking goes up to 1,500 hertz, right? So you have oscillations with lower frequencies, but when you go to higher frequencies, you have increase on the voltage of the cell, but it has no phase locking anymore, right? You have phase locking to the envelope of the sound, but not for the fine structure of the sound. And this is evidence for, this is consistent with the idea that we are computing only level differences in high frequencies, or taking some time difference, but for the envelope, not for the fine structure. And for lower frequencies, you can get both differences in level or time. And more recent results shows that you also compute level differences in low frequencies. This is the traditional ideas is that all low frequencies, time differences. High frequencies, only level differences. But now we see that level is for all, and time is most for low frequencies. Yeah. And thank you.
More questions?
Well, from what I know, is not the movement of the movement of the ears. They are changing mostly the level differences. Yes, so this is what happens in the owls, right? They have asymmetric ears. One ear that is faced up, one that is facing up. It's like a microphone to the up, other microphone to the right. So it's something like that. Then you have the level differences not so correlated with the ITD difference. For mammals that moves the ears, we move also a bit, but not as a cat, not. So the level differences would change a lot with the movement, but not the timing, right? The timing is more about the position and yeah.
More questions? Okay, Rodrigo, thank you very much.
Thank you very much.
We are pleased to introduce Dr. Aron Miranda from the Brain Institute. Today he will present Touch Without Primary Somatosensory Maps, Decision-Making at the Edge of the Sensory Loss. Let's welcome Dr. Aron.
Thank you. Thank you very much. Thank you. Good afternoon, everyone. I would like to thank the organizing committee for the organization of the event, Professors Claudio, Kerstin, and Tarcísio. So today I'm going to talk about a project that I started back in France during my last postdoc, and that I will continue to develop here in the Institute for the next years. So touch without primary somatosensory maps, decision-making at the edge of sensory loss. This title, it reflects our challenge to understand brain function by using the ablation behavior approach. So we started characterizing perceptual decision-making tasks in the previous lab. And by performing lesions with ibotenic acid, we have seen that mice, they still keep significant behavioral skills to complete the task. So we wanted to understand why. So our very initial goal is to characterize a behavioral task so that we can study the somatosensory function in more naturalistic settings. So our model to study perceptual decision making is the somatosensory system of the mouse that's especially well developed in these animals. The mice, they are nocturnal animals, and they use the tactile information as their main sensory modality. So they use whiskers actively. So they move the whiskers back and forth to understand the space around, to locate objects, and to both the spatial position and texture. The basic anatomy and physiology of the system is very well described. For example, all the sensory pathway from the primary sensory afferents through the brainstem, thalamus, up to the primary somatosensory cortex. This is very well described. We know the primary somatosensory, the somatosensory system is organized somatotopically, meaning it's organized according to the configuration of the body surface. Here, this is very sharply for the primary somatosensory cortex. This is also evident in the primary motor cortex and the secondary motor cortex, although less sharply, but still present. Our structure of interest is the whisker-related part of the primary somatosensory cortex. This is also known as barrel cortex. There is a one-to-one correspondence between the vibrissa of the mice and the cortical columns in the cortex. The reciprocal connections between the primary somatosensory cortex and the other structures, cortical structures, are well described as well. So this helps us to understand a more complex thing that is the relationship between the activity of these areas and the behavior. In one of the models that is used to understand what is going on during perceptual decisions, the sensory areas they integrate the sensory input, while the motor areas, they plan and organize the action. So while these motor areas, they continuously modulate the sensory areas with feedback connections. Now there is an accumulation of evidence process that is going on in the system until the decision is made. Now this model is a kind of a simplification. The thing is way more complex than that, but it's not necessarily wrong. It's just a simplification. We know that there is a lot of studies showing how the motor-related areas organize the actions, so how they plan and execute the actions, but how the primary somatosensory cortex is related to the perceptual decisions. This is not that much known, so our central question is how primary sensory areas contribute to perceptual decisions. Now, there is evidence for choice-related activity in the mouse somatosensory cortex. For example, in this paper, they used the go-no-go stimulus detection task. So here, in the go trials, there is a stimulation, and under stimulation, the mouse have to lick. So if the mouse, they don't lick, they miss the trial. In the no-go trials, there is no stimulation. If the mouse licks, it is a false alarm. Usually the animals are punished with more time for the next trial. And if they do not lick, this is the correct rejection. So in the study, they compared the responses of hit and miss trials in the primary sensory afferents, then in the VPM thalamus, that is the structures that project directly to the primary somatosensory cortex, and they see in both structures there is no difference between hit and miss trials. But in the primary somatosensory cortex, here through intracellular recordings, the responses for hit trials are way larger and sustained. So the activity is way more correlated with the behavior of the animal than with the stimulus. And what is interesting is that for the false alarm, meaning that it's in trials that there is no stimulus, And the animals, when they leak, there is also activity in significant activity in the primary somatosensory cortex. So this paper is just to show that there is evidence for choice-related activity in the somatosensory cortex, but there is something puzzling related to the activity of this region in perceptual decisions. For example, in this paper, in a similar task, mice, even after complete removal of the barrel cortex, they had just a transient impairment here in the first session. In the second session, they completely learned, they completely recovered the behavioral skills to the expert levels. So they can also learn the task at the same rate of the animals that were not lesioned. So this is lesion before the task learning. However, in another task, this time this is a gap crossing task where the animal needs to use the whiskers to locate a target platform that is located after a gap. So it usually is done in an environment that, or with in old experiments they used the blind rats. Nowadays we just use a dark chamber, and the animals cannot see the platform, but they use the whiskers for that. For crossing the gap. Lesions to the barrel cortex completely disrupt the behavior in this task. We just think that the barrel cortex function is critical for tasks that the perceptual demands are higher. So in this task, the difference, actually the main difference between they had fixed preparations, that is the previous task that I have shown, and the gap crossing is that in the gap crossing task, the animal needs to actively use his body actually to locate the platform. So they stretch the body to understand the distance between both platforms, and meaning this that the animal, they have to integrate head motions and body signals as well, proprioceptive signals and vestibular signals. And it has been described that this is important for active perception. And recently, there is work showing that there is a complete spatial navigation system in the primary somatosensory cortex with place cells, grid cells, border cells, everything that you find in the hippocampal formation, you can also find in the primary somatosensory cortex. So it's well possible that the barrel cortex lesioning disrupts way more than just tactile integration of tactile related information, but they also it can disrupt also the relationship with spatial signals around and head direction signals and so on. Okay, keep this in mind. There is another point that is this is a technical issue. We have shown it's common in the literature. Experimenters usually they trim all the whiskers in the face of on the face of the animal, letting just some few whiskers for the matter of control of the stimulation. This is just to make the experiment easier. You have less whiskers, and then you can detect. I mean, you can stimulate that specific whisker, and you also know the specific region in the barrel cortex that is related to that whisker. So this is important for recordings, for lesion, and so on. But we know this partial sensory deprivation induces cortical reorganization or cortical remapping. If we clip all the whiskers in the face of the animal, the correspondent barrel column, the activity of the neurons around the barrel column starts to expand. We say this is the expansion of the barrel. So this is due to more and more recruitment of cells around the barrel of the spiral vibrissa in the deprived barrels. So in this paper, this is a paper from the Gambino's lab where I worked. We have shown that the LTP is important for this process and also for the behavioral recovery during the single whisker experience. So just a short summary of what we have in the literature so far. So in the natural conditions, the animal normally learn and execute the task. In the single whisker condition, the animals also normally learn and execute the task. They just take a little bit more of time to cross the gap, but the accuracy is the same. But if we combine partial sensory deprivation and barocortex lesion, there is a complete impairment of the behavior. The animals, they cannot use the vibrissae at all to cross. We just think that this is a kind of catastrophic combination that the system cannot deal with two different kind of damage to this. Observe that you have a damage to the sensory apparatus and a damage in the barocortex. So that's why we think this result is common in the literature. And in the condition of natural whisker, the natural sensory condition and bilateral ablation, to our knowledge, there is no data in the literature. So we performed these experiments and we show that the animals, they can cross the gap. They can perform the gap when they are full whiskers in a good way, I would say. There is, the animals here in the last day, this is the fraction of success over session here for the learning phase. And in this case here, we just lesioned the animals after they were expert, and they get expert levels after 3 days. And here the animals were lesioned before task learning, and they could still learn. Even this guy that had almost 100% of the barocortex lesioned, he could learn. Here, this is color coded for the volume of barocortex lesion, right? So, and then just for a control, when we clip the whiskers, there is what we have seen in the literature. The animals cannot recover the behavior in this condition. So, we want to know what is the nature of this behavioral deficit, because this is probably not just to eliminate some interpretations. This is probably not a general somatosensory motor deficit or related to motivation, because full whisker mice, they can compensate for barocortex lesioning, right? So how do full whisker mice compensate for barocortex loss? This is our question. And why is an intact barocortex required for task performance under partial sensory deprivation? We think that this is probably related to cortical remapping. So our reformulated question would be, how does cortical remapping support behavioral skills recovery under partial sensory deprivation? So this is, we have, for this project, we have 3 key experimental fronts. Well, I developed part of them in the previous lab. I mean, the protocols. So we have the expertise for the characterization of the behavior. It's significantly advanced. The bilateral cortex lesion induction and quantification. We have protocols for developed and optimized protocols for that. And we also want to monitor single unit activity in the barocortex during the gap crossing behavior, especially during the single whisker condition, to trying to understand what is going on in the barocortex that allows for the animals to recover behavior in this condition. So starting with the behavioral characterization, this is the gap crossing task. It's a whisker dependent behavioral task. This is the schematics of the task. Here we put the animal in the home platform. There is an automatic door that when opens, the animal can have access to the gap area. The reward platform is movable, so we can adjust the distance. If the animal crosses the gap, he can get a reward. The equipment is, the apparatus is equipped with 3 cameras. Here it's a view of how the real apparatus looks like with the infrared sensors to detect the presence of the animal in the apparatus and activate and deactivate cameras, as well as the automatic food delivery. Here it's an example trial. When the door here it's open, the animal has access to the gap area, and then he can start to try to cross. He is usually protracting his body, trying to find the location. The challenge is to find the location of the platform in order to reach and grasp. and climb over the platform and then cross to get the reward. We use this machine learning based tracking tools like Deep Lab Cut to track the behavior of the mouse. The idea here is to understand what the animal is doing in order to cross the gap. We just see that this pretty much resembles the gap, the exploratory behavior of the rodents. So with several bouts of exploration, repeated bouts of exploration, and then this is just the y-axis position of the markers over time. So we can see the bouts of exploratory activity with the reach attempts, so we can quantify reaching attempts. We can understand what is the behavior, the behavioral outcome, actually. Because here, if the platform is close enough, the animal is going to reach for the platform. But if the platform is a bit far, they are going to use other strategies to cross the gap. So there are different behavioral outcomes associated with the different trials, with the different distance. So this is jump. They can jump if the platform is too far. So the animal completely goes off the platform. It takes off. And a lunge, that is something more like a horse gallop. He advanced with the two paws. So we set customized gap distance for the animals. Each animal is submitted to 4 levels of gap distance and catch trials, meaning that some trials there is no platform, just to know if the animal is performing impulsively. So here, the first and the second distance are more important for learning the task, for the animal to learn the task. This is not really whisker dependent, but the distance 3 and distance 4, these are important because distance 3, the animal can reach with the whiskers and with the 4 paws. We set the distance for that, for each animal. So the animal can reach with the whiskers and the 4 paws. So he can use a reach to cross, but the distance 4, he cannot reach. It's set for the animal not being allowed to reach for the platform, just with the whisker. So if we want to cross, he has to jump. This is an example. This is the data for one animal, where you see the trials and the gap distance that was set for each trial, with the color codes for the behavioral outcome from the trial. So they mostly use right and left reaches. I just separated here in yellow and blue, and lunges and jumps for more distance. This is important because if we want to understand the dependency of the behavior on the cortical structures, different behavioral actions must be considered. So not just a fraction of success or time or. So the first thing that we have seen was that the mice, they compensate for the barrel cortex loss by increasing reaching attempts. So here, they have the same graph that I showed in the beginning. In the third day, they have expert levels, but here you see that, well, there is a deficit because it's like they have less accurate reaches. They just need more and more reaches to cross. At some point, they are going to cross because this is a 2 minutes and 8 seconds trial, so it's a lot of time. This is for the animals that learn the task without the barrel cortex, and we see there is an increased reaching for this guy, especially because this is the guy with more part of the barrel cortex lesioned. And the compensatory strategy just collapse after partial sensory deprivation, so they don't reach anymore. This is the result for the guys that were learning the task without barrel cortex. So we clip the whisker, they don't use to reach anymore. So it's like they are not kind of trying to guess where the platform is. To study jumps, well, up to now, this is now a little bit complicated because actually we don't have that much jumpers in the batch. So there are some few jumpers. This is more or less half of the animals are jumpers, and not all the jumpers are consistent. So here we have just 2 animals that are consistent. Well, we can see this guy that was lesioned that took 40% head, but we cannot conclude on that yet. And regarding jump learning, we just saw that there are almost significantly here, but way less jumpers in the learning during the learning phase for the animals that were lesioned before. Here, just to show that there are some few these animals they did not have huge lesions. So what we can conclude up to now is that mice use distinct strategies for gap crossing. Strategy choice depends on how gap distance relates to each animal's biomechanical limits, meaning the reachings or the body stretch. In the multi-whisker conditions, mice compensate for the loss of barrel cortex by increasing the number of reach attempts, allowing for task learning and performance to persist despite lesion. And partial sensory deprivation collapse compensation in barrel cortex lesion in mice, revealing that this structure might be essential for flexibility and sensory motor relearning. Now, we just want to show our hypothesis. So considering that cortical remapping is required for behavioral skills recovery, and that mice also rely on head and whisker coordination strategies during goal-oriented active touch, and the recent discovery of a spatial navigation system in the somatosensory cortex with place cells, head direction cells, border cells, and grid cells. We think that during partial sensory deprivation, cortical remapping in the primary somatosensory cortex reestablishes the link between tactile input and head orientation signals. And these updated associations becomes essential for downstream circuits to plan and execute the actions. So in the perspectives, I just have two things to show: that we can track the head orientation and whisker angles. We can train models to track whiskers as well. And with this technique, we can track the head orientation. So it's something that are things that we are going to be implementing here in the next years. And also single unit recordings. We have the protocols, we have the expertise for that. So we just implanted some few animals. This is data that was not really analyzed in depth yet. But we have a way to record from animals performing the task. It's a bit difficult because they have to perform sometimes for the jumpers is a bit difficult. But there is one thing that is we can set is that we observed some jumpers jump more because of the food restriction levels. So if we control the food restriction to not to be that strong, we just keep the animals reaching for the task, so we are fine. Yeah, I would like to thank the team that I have worked with, especially Leah that did most of the experiments with me. Thank you for your attention.
Thank you, Aron. Questions?
So yes, and I see that now you're going to start tracking the whiskers, because I was going to ask if the lesions change in any way the movement of the whiskers.
Então, yes, we can track the actually we cannot track whisker deflection. We cannot see if there is whisker touch, but we can track what we can do with this tracking the way it is done. Is that we can track the oscillations, just the oscillation. We cannot do more than that, because the markers are not really very precise, let's say. See, and it's it's an information that we can use for that to know if there is a change. We can analyze that. I didn't do anything related to that, but yeah, we can look at that. There is the paper that I showed in the beginning. That the animals they recover the behavior. Yes, they see that there is there is an effect of the lesion on the whisker movements, but still the animals, they can find a strategy to
The other thing that I cannot help thinking is like when I look at the rat's whisking, it's like it's crazy, right? The complexity of the movements and such. And also you look at the system, you look at the complexity of the barrel cortex, and for me this means like, wow, this system must do some like crazy coding of patterns and shapes and So essentially your task has one bit of information, right? Like is there a floor nearby or not, right?
Yes, but the things that we plan to actually, yes, I agree, because this is that something that we can vary the perceptual demands of the task, I think, because in the configuration now is that we have a huge platform, so that's why the animals, they can reach several times, and at some point they are going to find the platform by reaching. But we can, and I did some pilot experiments with putting a kind of a support on the edge of the platform and kind of adjusting. So the mice, they have to target that specific place.
Yeah, but still, would you say that this is not a challenge like for the whisker sensory system? Okay, it's harder to find the thing. So it's like about like spatial search strategies. But finding it or not, sensory-wise, for the whiskers is one bit of information still, right? So I'm thinking it's like if you want to see the effects of lesion in the barrel cortex, aren't there like other tasks that fully explode the coding capabilities
with
of the
textures, you mean?
like textile shape, something weird?
I don't know. In the way it is already a lot difficult to those experiments. I mean, the probability that the experiments goes wrong.
Yeah, I know.
Yeah, because if, for example, if the animals follow the platform and he knows the apparatus below, you have to forget, and then
I know it's a pain to train these guys. Yeah, I know. But because actually I would think like even if you cut the communication from the actual whisker sensory system, maybe they will feel touching the other side. We were talking the other day, like just from deflections of the skin or whatever, like other trigeminal pathways, right?
I don't know. I don't know if there is a way to calibrate for that. Yeah.
I'm just saying I don't see this like recruiting, demanding too much of the barrel cortex, right? This task, I don't see it demanding too much of the barrel cortex. And that's my intuition of it. But
I don't know. I didn't get it.
So, yeah. So this stuff about if you have even if you remove completely the S1, you still have some feelings or something coming from the trigeminal. But even if removing, have you considered, for instance, the other pathways, for instance, the thalamus to secondary somatosensory areas, similar to what we discussed before, like the blind side in the visual areas, that there are some lesions in the primary visual cortex. The subject says that cannot see the object, but can perform some visual tasks like reaching and grasping.
So, but I didn't get the question in the end.
So if
you lesion the
so
other,
if you have
other
the influence
structures
more than just having the primary, so other skin stuff, but also information from higher somatosensory areas influencing the discrimination.
Well, what I understood from the literature is that if you the barrel cortex for detection, I mean if you don't have the necessity of the spatial localization of the stimulus, the barrel cortex can it doesn't have a crucial computational contribution to the system. You see, it's like the system can reorganize and can be used can use information with less complexity to decide, like stimulation or no stimulation. This is pretty far from a condition that you require for the system to know the spatial position, you see. There are experiments with lesions to the it's not to the thalamus, I forgot now, it's a pathway that is going through the hypothalamus and goes to the cerebellum. There is a paper that shows that if you lesion just it's a small spot, okay, the animal cannot behave as well. And also in the VPM thalamus. But this is also structures that are in the thalamus. There is structures coming from the primary motor areas, for the primary and secondary motor areas. Yeah, I don't know. They prefer to reach. Yeah, they prefer to reach for the shorter distance. Yeah, they prefer to reach for the shorter distance here. I mean, the reaching is the most economic decision, right? They are not going to jump if the but as soon as the distance are getting more challenging, they jump or they lunge. The lunge is a bit different because the animal they do not get that much impulse. They just go with both. No, we didn't. I forgot to say, but in the quantification there, we put together this kind of more dynamic action. I just put lunges and jumps. I consider it the same. Yeah, we don't have actually that much data to analyze between jumpers and in jumping and in the different actions.
Can they use?
No, because this is a dark chambered box. This is light proof. Well, in the beginning, I mean, speaking about other sensory modalities, we had a lot to do regarding troubleshooting of this task because the animals they could cross. When I arrived at the lab, they were doing a paper, and the animals. I mean, they were just saying the animals were jumping. No, then I observed they were actually reaching, and they were saying this is a whisker-dependent behavioral task. And okay, let's do an experiment that is just clipping all the whiskers of the animal and submit the animals to the task, and the animals were still crossing. And so there is something more. And then we realized the animals could use the smell of the platform to cross. They can use the smell of the platform. They could use at some point the noise from the food delivery system that was doing a small noise. So even that we removed completely the platform, they were jumping from the home platform until the other base was really far. So we had a lot to do, and now we know that those platforms have to be cutted using water jet. It cannot be cutted with laser because then this produces a smell of burned stuff. And the apparatus needs to be okay regarding noise. They can use other sensory cues, but not vision.
We have one more question. Okay.
Yeah, I was wondering if this rewiring that you are talking about could be happening in the thalamus instead, right?
It can happen in the thalamus, but the extension that it happens to the cortex is when there is some modification in the thalamus. I don't remember exactly now. I have seen a paper talking about that, but I don't remember. Sorry, I can look for it. But the cortex is the structure that is that the plasticity is stronger, right? So it's no. This is this data. That is data. Sorry, this guy is not. I'll put in the slide. Yep. So yeah. This is the expansion of the barrel we see with intrinsic optical imaging, right? Well, with electrophysiology, I didn't have seen that much things, but what Tiago did when I arrived in the lab, he was working on it. He was doing patch clamp actually, and they could see that the AMPA receptors diffusion surface of AMPA receptor was important for that. That's why this was the technique they used to kind of to block the synaptic plasticity. They had an AMPA cross-linking approach. They used antibodies for that. But yeah, the plasticity they can see with intracellular recordings, so they can see after. There's something that I don't understand that much, but they after the barrel is expanded, when they make recordings of the layer 2-3 neurons, they see that the LTP is already saturated, let's say. But yeah, the expansion of the barrel, if that was your question, if the expansion of the barrel is seen through the intrinsic optical imaging, we don't use electrophysiology for that. I just think that we need a lot of something if we want to use electrophysiology to see the barrel expansion. But this is really well described. It's something that it's happening for sure in the whisker, in the single whisker condition. Mm-hmm. Yes, there are. I didn't read that much these papers, but there is a Michael Messernick papers, but I don't know if they use the. They use the electrophysiology. Kevin Fox, yes, Kevin Fox, yes. It's single unit. Yeah, I know. I have seen, but I don't remember exactly what was his finding. Yes. Okay, thank you.
Okay.
Mm. Mm. Through the POM, you mean? Uh-huh. I see. Yeah, curious. Mm-hmm. You have seen some data showing that, no? Okay. Nice.
Okay, thank you, Aron. Okay, so the next talk is going to be held by Dr. Joaquin, is that? Joaquin Gonzalez from the Brain Institute and NYU. Today he will present subspace communication for the hippocampal-retrosplenial axis. Please join me to welcome Dr. Gonzalez.
Okay, thanks for inviting me to give this talk. I'm going to tell you about the progress I made after joining the Chen and Bushaki lab this February. This talk is going to be about communication between hippocampus and neocortex and how this influences, or how we believe this may influence memory consolidation. So we know that the hippocampus and neocortex need to interact in order to consolidate experience into long-term memories. That's something we have known for at least 50 or 60 years now. You can think about, for instance, patient HM. But the truth is that the current model that we have, which is the system consolidation theory, which states that there is a fast plasticity site in the hippocampus, and then this information or these associations are transferred to the neocortex. It's kind of an empty model in a sense that we kind of don't understand what information is actually being transferred to the cortex, or what are the mechanisms that allow this exchange of information. So we can take kind of this population view of neuronal communication, and this population view basically kind of stems from these current devices that we have to record populations of neurons across different areas, and the way that we can analyze these data sets. And we know that this, we can think in general terms that communication between brain regions is determined both by interregional connectivity and local network dynamics, local neural dynamics. And there is kind of an open problem still that we don't know exactly how one brain region transforms its inputs into its outputs. And this is an open problem that I will try to address at least to some degree. So we need to take a kind of mathematical detour, but I will try to make it as intuitive as possible. And the idea is that we can think about neuronal communication as trying to find these subspaces. This word will come a lot in this talk, which is basically the following. So we know that if we have recordings between two brain regions, we know that only a subset of neurons will project from region 1 to region 2, for instance. So the idea is, for instance, if you imagine that you have like neuron 1, neuron 2, and neuron 3, this defines, we can think about the population activity as being just at trajectories in this high dimensional space. This illustration shows just 3 neurons, but we can think about this having as many neurons as a given region, and then each one of these axes will be the firing rate of each neuron. We can make much more complicated models, but this will kind of give you the basic overview. And so now suppose that like neuron 1 and neuron 2 project to this downstream area, but neuron 3 does not. So in a sense, all the type of population patterns that somehow show variations in this axis and this other axis will be transmitted to this downstream population, but neuron 3 can do whatever he wants, and it will never be read by the downstream population. So in a sense, this idea of understanding communication between populations boils down to finding these low-dimensional subspaces, and the idea of subspace, you have the full space would be just like a cube that covers this entire volume, and a subspace would be some volume, sub-volume of this kind of cube. In this case, this simple example shows a plane, and because only these 2 neurons are the ones projecting downstream, while this other neuron does not project downstream. And this finding of these subspaces, we can also think about them in terms of trying to find a set of weights. So a set of weights for each of these individual neurons in a way that if we generate a linear combination of this set of weights times the activity of each neuron in each brain region, and we also have a corresponding set of weights for the downstream population. When we generate this linear combination, we get the maximum amount of correlation between these 2 brain regions. This is in a general sense. These are ideas that are originally developed in this 2018 paper, and sort of the field has evolved towards this idea. What's kind of new is that so you can find these subspaces if you have 2 recorded regions simultaneously. If you have more than 2 recorded regions simultaneously, you can start thinking about what happens if we record another region, how the new subspace is related to the original subspace. And for instance, you can think now if the only neuron 2 and neuron 3 project to the downstream region, then only their variations of their patterns will be projected, and neuron 1 will do nothing. So then the input-output transformation of this circuit is basically a mapping that transforms this input subspace. In this case, for instance, the subspace spanned between CA1 and CA3 to an output subspace, in this case with retrosplenial cortex. And again, we have this also set of weights for each individual neuron, and we can try to understand how these 2 subspaces are related, and this will give us a prompt or an idea of how this input-output transformation is taking place between these 2 regions. So in this case, we had these awake recordings in the hippocampus and neocortex. So these were performed in the Bushaki lab by postdoc Mishi Borlaskos. And he recorded these two type of different experiments. In the first experiment, he had these synapse recordings. So this is a new type of probe that has 1,000 channels that can record simultaneously. And this is kind of was developed in order to cover the entire transverse axis of the hippocampus. So we basically can record neurons from dentate gyros, CA3, CA2, and CA1. Kind of the drawback of this probe is that it's rigid probe, so we need to record these animals in head fixed conditions. And throughout this talk, these animals will be recorded head fixed, but running on a treadmill, and we will just use the high theta epochs. As you can see, we can record unit activity from each of these regions. And we also had these other recordings that he made with these NeuroPixel 2.0 probes. So you probably know about these ones. And the great advantage of the NeuroPixel technology is that it's basically very reconfigurable. So meaning we can maximize, it has many channels, but we can record from 384 channels simultaneously. And we can sort of maximize the number of channels to cover retrosplenial cortex, for instance, CA1 cortex, CA1 region, and CA3 of the hippocampus. And the idea is the following. We will take CA1 to be kind of the main protagonist of the story, being the output node of the hippocampus. And we will take CA3. CA3 is known to project. It's kind of like one of the most studied synapses in the brain, CA3 to CA1. And then we'll take retrosplenial cortex to be kind of a downstream target of the hippocampus. We know that CA1 needs to project through subiculum to then reach retrosplenial cortex, but we know ripples, for instance, propagate to retrosplenial cortex. We know retrosplenial cortex shows a spatial map inside. And the advantage of this probe is that it allows us to record in freely moving animals, for instance, animals that are slightly water deprived, and then they do a task in which they run back and forth through a noble maze to get water rewards at both ends. So we can take this idea that I told you about subspaces and try to quantify them. Basically, this is kind of similar to doing a principal component analysis, if you're familiar, but instead of doing it on a correlation matrix, you do it on a cross-covariance matrix that has the activity or has the correlations of the different regions inside. And then we can try to relate how the weights, for instance, of one subspace, let's say how the weights of the input subspace are related to the weights of the output subspace for each individual neuron, and then we can study kind of more complex relationships that these two subspaces may have between them. So the first kind of result that we got, or the first thing that I tried to do is, okay, I quantify the number of significant subspace using surrogate methods. This is kind of similar again to the cell assembly detection protocols that Adriano and Vitor have developed a long time ago. And the idea is that we find for each neuron, each one of these dots will be a single neuron weight, and we will try for the neurons in CA1 to relate how for instance, the weights, the absolute magnitude of the weights between the CA1 subspace with CA2, how they are related to the CA1 weights with the subspace related to CA3. So meaning two areas that project together, or we can think, for instance, so we have here, for instance, how the weights of CA1 related to the inputs and the CA1 related to the outputs are related. And we found that overall there is a pretty consistent correlation in the subspace weight magnitude. So meaning that if a given cell will be featured strongly on a given subspace, so if a given cell communicates strongly on one pathway, it's likely to communicate strongly on another pathway as well. And this was very correlated across all sessions and these two type of recording probes. So meaning we have some cells that show this strong contribution, some other ones that show less strong one. But we can think, so this kind of view kind of gives us the sense that the same cells that mediate the inputs are also somehow responsible for the outputs. And to my mind, it was kind of a bit of an odd result, because why would you put a brain region there if you can't just lay down an axon, right? Just put an axon and the axon will do the same as this kind of case. So I thought, okay, maybe there is something a little bit more complex going on. Let's try to understand how these subspaces are related to each other. So going back, we try to measure, or I try to measure how much the input subspace, what's the angle between the input subspace and the output subspace. And so, for instance, you can here see kind of a visual representation. For instance, this would be the first dimension, or the first one of these axes in this input subspace. This one would be the weights. Each one of these dots is one single CA1 pyramidal cell. And then we have this other axis as well here. And then this is the same for the output subspace. And you can see that overall, again, this if a cell has a strong, and the thickness of the line speaks about the weight of the magnitude of the weight. And you see that even though, for instance, this cell here is strongly featured for the input subspace and the output subspace, you can see that it's very strongly featured in the most important input subspace, but it's kind of not featured in this other subspace as well, but it gets kind of displaced to this second one. So you can see that overall this is not a mirror image. The input and the output is just not a mirror image, but instead it has some type of rotation that's taking place in this high dimensional space. And what I did was to quantify what's called principal angles. So you can think, if you have two lines, these two lines determine a single angle, right? But if you have two planes, these planes determine, the two planes determine two different angles, and if you have n-dimensional subspaces, they determine n-dimensional angles. This is called principal angles or Jordan angles, if you want just to Google them. And the idea is that these have some set of properties. For instance, if the maximum angle equals pi over 2, it means that at least there is two dimensions that are orthogonal one from each other, this kind of properties. So we computed these angles between the input and the output subspaces, but we needed somehow a kind of a control to understand whether whatever value of an angle, what it meant. So what I did is the following. So I computed the angle, for instance, between the CA1-CA3 and the CA1 retrosplenial subspace, and I compared that angle to basically computing the CA1-CA3 subspace, but in different moments in time, right? And this should give us a sense of how much rotations are just due to just random fluctuations or non-stationarities in the recordings, or like the emergence of place fields and the disappearance of other place fields. So I basically computed these angles within the same subspaces and across different subspaces. And what I found is that basically this input-output transformation boils down to these different subspaces being rotated one from another above chance levels, right? So it means that even though the same neurons are kind of mediating the inputs and the outputs, kind of the combination of which neurons are coactive are changing between these two conditions. And we also find that the degree of rotations within the subspaces kind of depended on the brain region. We found that CA1 was the region that showed the strongest or the largest kind of rotations. So this shows that we can transform inputs into outputs. Oh, whoops. Okay. We can transform inputs into outputs by rotating one subspace with respect to the other. And this is kind of a default brain operation that takes place when transforming information and broadcasting information from one stage to the other. And just to give a sense that this kind of subspace rotation is kind of a general principle or a general solution for transforming inputs into outputs, I did the following. So I trained a recurrent neural network, so an artificial neural network, to transform the activity of CA3 into the activity in retrosplenial. So basically, I'm providing the artificial neural network the activity of CA3, and I'm making the network or training the network to predict the activity of the retrosplenial cortex. So this is kind of the inputs you get to the give to the network, and this is the outputs that the network should produce. And if you see, there is not kind of a non-trivial relationship with how to transform this time series into this other time series. So it's not that the input is completely simple. It's not that these are like sine waves and you just need basically to copy one to the other. But there are more complex relationships. This is kind of what brought some problems to this initial analysis of these multi-regional interactions. But we know that, but we found that this vanilla recurrent neural network is able to predict the activity of the retrosplenial cortex based on CA3 activity. So it's kind of, it's able to reproduce what CA1 is supposed to do. So it gets a good accuracy in the test data set. And the idea is how this network able to transform inputs into outputs? Well, it's able to either change its internal connections, or it's also able to change its input subspace and its output subspace, because the way that these neural networks, these recurrent neural networks work, at least the very simple ones, is that the inputs are a linear combination that's provided to the neurons in the hidden layer, and the outputs are read basically as another linear combination from these hidden neurons, right? And what we found is that we compare what happens before training and after training. We find that there is a consistent rotation that takes place with learning. So basically transforming the inputs into the outputs somehow depends on rotating these two subspaces, the input and the output subspace, one with respect to the other. Okay, so what's the meaning of this subspace rotation that we studied and we saw in spontaneous conditions? To get an idea of what's the possible meaning that this rotation may play, we had this experiment that Mishi performed in the lab, very nice experiment in which we have the animals going, these two mazes are known, so the animals go first maze 1 and they perform a set of trials, again getting rewards at both ends. These are mice running back and forth. And then in a very brief short time, we take the animals out and we replace the maze by a different type of maze. In this case, can change from a metal linear maze to a wooden maze with a different size. And we open up the enclosure. So it's basically a different context. And we make the animals run the same set of trials. And the nice thing about this experiment is that we have this neuropixel recording, so we can track the activity of these three neuronal populations, CE3, CE1, and retrosplenial, the exact same neurons across these two different contexts, because this is basically the same session. And we carry the subspace analysis. I'm not going to show. This is just basically computing one subspace and projecting it over time. And this is kind of the representation that we got when we compared, for instance, the subspaces for the first maze in retrosplenial cortex, for instance, and the second maze. And what we saw by and large, and if you kind of examine this plot, you will realize is that the individual weights of the cells were conserved, again, kind of mimicking the thing that we saw previously. So meaning if a given cell in either each of these three regions was going to be featured strongly on one maze, it will also be featured strongly on the other maze as well. So in that case, the subspace memberships kind of generalize between different contexts. But what we found is that if you compute the subspace in one maze and then project it to the other, remember this is just a linear combination, I can take this linear combination and then project it back to the second maze. What we found is that the activity between different mazes, so how much correlations the subspace in maze 1 explain in maze 2 basically drop to 0. So it means it doesn't perform above chance. And sort of our control in this case was we had these longer sessions in which we just split it in half. So the same maze, longer sessions, we split it in half. And if the maze remains the same, so the context remains unchanged, in that case, the subspace do generalize. But if we change conditions, the subspace kind of break. And we studied how these subspaces are related, or the memberships of these subspaces are related to their physiology and anatomy of the cells. And in this case, the nice approach that we have is that this is a very linear method in which each neuron will be assigned a given weight. So we can define, for instance, members of a subspace and non-members of the subspace by just using some thresholds on their individual weights. And what we found is that across these three regions, subspace members tended to fire at higher rates, even during pre-maze non-REM sleep. So before the animal ever encounters this novel maze, subspace members already fire faster and showed more burst activity. And they also showed a differential laminar profile. So across these three different brain regions, for instance, in CA1, this subspace member tended to be located more deeper within the CA1 cell layer. They also tended to be located deeper in CA3. This is now, if you follow kind of this literature, these are cells that are also called athorny cells. And also in retrosplenial cortex, we also saw a bias towards deeper layers in the cortex. Kind of to get an idea of what's the functional, so why are some cells featured more strongly? What we found is that these cells that communicate more strongly also tend to represent space in a population code in a stronger way. So this is to do this analysis, but we could kind of do the same thing with the traditional place field analysis. We computed these manifolds using UMAP, kind of a popular method nowadays. And what you see here is that it's kind of a low-dimensional view of the hippocampal activity. And this is color-coded by position of the animal or by the trial number in which the animal is placed in. And you can see basically that the population basically shows a geometry that is kind of similar to the maze. It's kind of a circle because forward and backward runs kind of involve different populations. So this is basically a circular maze in some sense. And we see that the subspace members tend to capture much more of this geometry than non-member cells. And finally, to get an idea of whether these subspaces play any role for learning, we basically measure whether these subspaces will be reactivated during post-experience sleep. So these animals went through a novel maze learning, and we projected the subspaces computed during the maze runs in the pre-maze sleep and the post-maze sleep. And what we found is in large, both the CE1, CE3 subspace and CE1 retrosplenial subspace, they both get reactivated more strongly during post-maze sleep compared to pre-maze sleep. But kind of the interesting pattern started emerging here, in which we saw that the CE1, CE3 subspace will be reactivated particularly during ripples, but the CE1 retrosplenial subspace will not show kind of this pattern. This kind of prompt us to think, okay, maybe there is some difference also in the type of content that these subspaces are being reactivated. So we carry out the replay analysis, in which we basically try to decode the position of the animal during these population events during sleep. And what we found is that when the CE1, CE3 subspace reactivates, you can see here it gets a large correlation of 0.4. We see an event that very much resembles a trajectory of that the mouse made during the runs. This is called a replay event. When the C1, C3 subspace is not reactivated, we just see basic random jumps that don't resemble anything. But interestingly enough, when we saw that when the C1 retrosplenial, so the output subspace would get reactivated, there was no increase in replay, and the replay actually increased when the C1 retrosplenial subspace would not be reactivated. So we see this duality in which we find a plastic subspace related to the input, the inputs of C3, and we see a more rigid and stable subspace related to the output to the cortex. And this also, we verify this kind of similar profile using cross-correlation between the spike times during this pre and post-sleep. And what we found is that if we take the activity, so the time 0 will be the spike of a C3 neuron. We see, and then we measure the cross-correlation. We see that during post-maze sleep, in orange here, there is an increase in the spike transmission probability taking place so it's taking place, there's an increase in the spike transmission probability for cells that belong to the CE1, CE3 subspace. But when we measure the same thing, but for cells that belong to the CE1 retrosplenial subspace, so in this case, time 0 would be the spike of a CE1 neuron, we see that these cells tend to decrease their activity. And what we find is kind of this dual plasticity rule in which the members of the CE1, CE3 subspace would get potentiated during post-maze sleep, but the members of the retrosplenial subspace wouldn't show any change at all during post-maze sleep. And what we found was the opposite, that the correlation between CE1 members and retrosplenial non-members, so there was kind of a decrease correlation taking place, which kind of suggests that there is some depression that may take place in this other subspace, in the output subspace. So kind of to get some conclusions and discussion, we see that there is a fraction of non-random neurons that communicate across different contexts, and we kind of think that these are deep cells in CE1, and they are athorny cells in CE3, and they are deeper cells in the cortex. So we have this anatomically defined communication subspace across these 3 different regions. We also found that this is combinatorial code that can give rise to different communication patterns in a context-dependent way. And kind of the message is that if you are bounded to use the same set of cells for different tasks, the way that you can introduce novelty is by rearranging the combinations of them. It's kind of similar to the way that, for instance, if you need to do different recipes with the same set of ingredients, you can basically recombine the way that you can rearrange the way that you combine these ingredients to generate different kind of dishes and generate more novelty that way. And then we also find that there is kind of this balance between plasticity and stability, kind of reproduces what we know in the sense that the intrahippocampal subspaces, they look more plastic, and we kind of see a type of plasticity rule that would be more similar to LTP, while the communication between hippocampus and cortex is kind of more rigid, and the type of plasticity rule we see is more reminiscent of long-term depression. So we see this balance between the two of them. So related, I think this is an important code, the important part of the paper, that we will release basically all this data set in a format that is suitable for people to use it. and employ it freely. And we will also release the codes in order to measure this subspace computation. I didn't have too much time to go into the technical details, but this is an addition over normal methods that is allowed that allows to condition on third region measurements in order to get more precise correlations. And basically, I want to acknowledge, first of all, Mishi, who performed all the experiments and basically gave me this data set to play with. And then my two supervisors, Sage Chen and Yuri Bushaki in NYU, for supporting me. And thank you for listening to me.
Thank you, Joaquin. Questions?
Hi, thanks, Joaquin. I have a question related to the terminology, because I mean, as far as I understood, we are looking to the weights, because when you have CA1, CA3, and CA1 retrosplenial cortex, you are always analyzing the weights at CA1, right?
Yes.
You don't use the other regions, I mean, to compute the weights, but
No,
then
I
you are...
mean, I did have the weights in the other regions, and we did do some stuff with it, but for the sake of simplicity, I didn't show too much.
So my question would be, why don't you simply say that the subspace, they differ, or why do you use the word rotates? You know, I
Okay,
mean,
because I mean, they
would-
actually not, okay, so they are actually not, depends on how you think, so how do you say that they are different? Because when you measure the individual contributions, so you measure the absolute weights of the given cells, they are very correlated across all different subspaces. This is kind of what I'm showing here, and this is the
Yeah,
first
they
approach
are-
I took.
I mean, they are not that strong correlated, but they are correlated.
They are 0.7 almost. I mean, we never see this type of correlations in neuroscience, right? I mean, correlations are
Well,
very weak,
okay.
usually. But I mean, yeah, of course, they are not perfectly correlated, so there is some regression to the mean problems, and there are some things that you can argue there. But I think the idea is also that kind of on a first principle basis, I think the way that you need to understand and think about these subspaces in terms of their geometry, this is kind of like the most
I mean,
natural way of thinking
another
about
way
them.
of framing my question is that can you have two different subspaces which are not rotated from each other? Is there such a thing as they are not being rotated? I'm just asking
no.
the
No,
jargon.
yeah. I think that the idea of being rotated and then being different is kind of the same. Yeah.
And then another question is, because when you say rotates, gives the idea of something dynamically happening, but in a way,
Yes.
so you have static subspaces.
Yes.
It's not that
Yes.
they are rotating to fulfill
Yeah.
some needs. They are always there. Right?
Yeah, exactly. Yeah.
And then, I mean, I can let other people make questions.
Thank you.
I have a philosophical question. Joaquin, what's the nature of a computation for you? Do you think that your method is a way of describing what is a neuronal computation, or do you think it sheds light to the nature of the computation itself? You understand? Because for us, if you have the input and the output, what happens in between is what we call today a computation.
Yeah, exactly. Yeah, exactly. That's the.
And you see this in the work of David Ma.
Yeah.
And you could, in principle, apply your method to any place.
Yes.
Or so it then touches a very important aspect in the nature of computation. So my question is, do you think your method is a simple way of describing it or characterizing or quantifying it? Or do you think it sheds light to the nature of what a neuronal computation is?
Yeah, that's kind of a deep question. I think for the my first approach would be to say it's kind of a description, but I think it can shed some light in terms of the nature of the computation. So, for instance, we find a difference between the amount of rotations we see, even when we match. So, for this panel, we match the number of neurons in CA1, CA3, and retrosplenial cortex, in order for the neuron number not to be a bias in terms of the measurement that we are having, that we are obtaining. And what we find is that there is different amounts of, so for instance, it looks like CA1 is the region that it's more able to rotate one subspace with another, so meaning that representations are more orthogonal in CA1 compared to the other regions. And in a sense, you can think about, okay, so if representations can be more orthogonal between each other, it means that the dimensionality of the CA1 code it's larger compared to these other types of codes. And then you can make a parallel with, for instance, I don't know, like artificial neural networks in which like an autoencoder, which you start with a very low dimensional signal, you expand the dimensions, and then you contract the dimensions back, and then you autoencode but you're able to just somehow transform using this high dimensional space, and that somehow it's related to the computation that is taking place. Maybe when you are thinking about computation, you're thinking pattern completion, pattern separation, maybe. And in that sense, also you see that, for instance, CA1 shows a larger dimensionality, so in a principle it will be allowed to separate more kind of patterns, and that may be related to the kind of computations. I think, I mean, we discussed this with Aron a long time, like what do we mean actually by computation is kind of a difficult thing in the field, right? I think it's kind of a mixture of both. I cannot completely state one or the other. I think the method is a description, but through the method we can get an idea, at least a very simple idea, because as Adriano said, for instance, I'm not seeing these rotations develop over time, for instance. That's kind of the first thing I thought when I started computing these things. Yeah, so it's kind of, I think we can get some insights into what the computation is. For instance, the fact that we see some more plastic and more rigid subspaces, I think also relates to the kind of computations that the input and the output it's doing. We know that intrahippocampal communication is kind of a fast plasticity place in which associations develop rapidly. The cortex, then it takes more time, even can take weeks in order for information to be passed, even though we don't know exactly what's the nature of the information being transmitted to cortex. Yeah, but I would say maybe in a few years I can get a more complete answer for this. Yes. What do we learn? Yeah, I think yeah, that's a kind of, I mean, for sure I'm not going to claim that I solved systems consolidation, right? I think this is yet but a step towards doing that, in the sense that we have a large data set and we kind of deal with this complex problem of understanding how these three related populations interact with respect to each other. Laying down this foundation may allow us to go a little bit farther than we know thus far. We see that the CA1 somehow balances some fast changes that take place within hippocampus with this slower sending of information back. And I think there is a possibility that that even within CA1 there are maybe several plasticity rules that take place. So meaning that LTP may develop during CA1, CA3 communication, but then the kind of plasticity rule may change towards cortex. So then you have more like a long-term depression that's taking place at that interface. I would say that's kind of the value for now for that big story. I just wanted to frame what's the importance for the communication, but for sure this is, I think, like a baby steps towards that goal. But nonetheless, this has been kind of a complex issue, how to relate. For instance, when you have three related populations, it's kind of non-trivial to understand how they are related one to another.
I have a similar question. Actually, I have two, a technical and then conceptual question. Maybe I start with the technical. So covariance analysis in very high dimension is statistically a very tricky thing, because your data sample may not fill the space. And then you have spurious principal components. So I would like to understand how you deal with this problem in this analysis.
So I didn't show, but I mean, the way that I deal with this, so not to think about, I mostly draw this kind of geometrical analogies just for the sake of visualizations. I think they look nice, and people kind of, that's the way that they present this data. But the way that I did the following, so you find a linear combination that maximizes correlation. You make sure that the amount of the data, so the length of the time series is long enough compared to the number of neurons you have in each recorded region. So I would say we typically have around 80 neurons per brain region, but the length of the data set in which we are computing these correlations, maybe it's well beyond, we are recording like maybe around 2-3 hours, and we are sampling this, so the sampling rates we are using, it's around 10 milliseconds, or even, it's 1 millisecond, and we have some smoothing going on. So once we have this, that covers the terms of the statistics, so we are confident that we can use in this case, canonical correlation analysis to compute and get some meaningful value. But then what we do is I do the following. So I do a surrogate control in order to see how many components are significant. So I start shifting one of the two time series with respect to the other and compute canonical correlation. I get one correlation value. That correlation value is kind of the best that you get out of a spurious correlation. Out of that you construct a null distribution, and then you see how your actual values, correlation values, were related to that. And we typically see that, so you, with canonical correlation, you get the number of components is equal to the minimum number of neurons you get in the two brain regions. So say we have 50 components, we typically get 5 to 10 components maybe at top that show some significance. Then we also had another verification that when you use the canonical, the weights, the CCA weights, and then you compute kind of an explained variance for each region, you do see that the first component, so the one that accounts for the most correlation, also explains more variance, so it means that it's more important locally. But this is kind of the controls that we are making. We also downsample finding rates to see that some finding rates are not an issue here. Yeah, that's more or less around the...
Yeah, thank you. So for the conceptual one, so your basic motivation in the beginning is systems consolidation.
Yeah.
It must somehow happen. Nobody knows really what is going on. So at the end of systems consolidation, more of that memory trace or the ability to do pattern completion, more of a type of operation should reside at the cortical level. So how is this tool set suited to detect that kind of transfer is a question. And so if I did get it right, I'm not sure. Yeah? So there's in your retrosplenial set of neurons a subset that's not so much communicating. Yeah. So one hypothesis would be that that's exactly the neurons that could do the job in the long term at the cortical level, because why would you use the strongly coupled population that is always seeing the hippocampal input to then store at the cortical level? So they are all one synapse away from the projection population. So how do you think about systems consolidation, the memory engram then ending up at the cortical level, and maybe it would need other tools to really do this, because you are, as you said, looking at covariances, projections, linear operations between these regions.
Yeah, I think more than a tool shortage, what we have is a data shortage. I think that the closest thing that we had, and I had some discussions with I think what we miss in this case, but of course it's a kind of a very technically difficult thing, is to have these recordings over long term, maybe 2 weeks, 3 weeks. I discussed some things with the Kim paper by the Ganguly lab, that they show that there is this development of intracortical synchronization that takes place during rich learning, kind of controversial because it's kind of a procedural thing, but hippocampal dependent. But they showed this progress that takes place around 7 days at least for for kind of computations to be independent of the hippocampus and to be dependent of intracortical operations. So I think if we could record this activity during a longer time period, we would get a better idea. I think that the tools kind of would be there, because I mean it's kind of, my idea would be that yes, that's right. Yeah, but I think, I mean, first of all, you should establish the amount of representation drift that takes place during that time. After dealing with that and seeing what's the amount of representation drift, maybe you find that I mean, I would say the likely thing is that the cortex would show less representation drift compared to hippocampus. Yeah, and then basically doing this kind of same set of tools, but just yeah, I think it's a what? Oh, representation drift is the idea that representation change over time. So I think the most clear example is in olfactory cortex. So you see that the neurons that encode, so there is this paper by Carl Schonover in the Richard Axel lab, and they see that, so if you record neurons through 30 days, for instance, you see that the neurons that encode a given order in day 1, they are not the same neurons that encode that same order in neuron 14, in day 14 or something. And in the hippocampus, it's kind of the same. You see this change in the place fields. This is kind of analogous to the representation drift. So you see that a given cell that has a place field and over a set of trials and then all of a sudden that place field disappears or another one appears. And so there is a kind of a technical problem as we are discussing of, because over long-term periods, it's likely that the hippocampal representations will change. I don't know if it's the subspace memberships will change as much. I don't know if the CE1, CE3 will change much. Maybe the CE1 retrosplenial then becomes a little bit more stable. I think, I mean, I would love to analyze that kind of that data set, but I think that's the critical aspect would be to get the 14 days straight more than the method. The method can then be fitted to the data, but the data would be for sure the key.
Okay, thank you very much. Any more questions? No? Okay, let's thank Joaquin.